Our first presentation is from Dr. Sudeep Popat, who is an associate professor in the Department of Environmental Engineering and Earth Sciences at Clemson University. His group focuses on wastewater treatment technologies, particularly anaerobic digestion to recover energy and electrochemical technologies to recover nutrients. Dr. Popat has published over 45 peer-reviewed articles and book chapters and has been involved in projects funded over $22 million, and his share being $2.5 million. Very excited to have you, Sudeep. Take it away.
Right. Thank you so much. It's a pleasure to present again. I was just reflecting back on when I first presented at the summit, and it was about, I think, 5 years back, and I think I may have presented every year. So it's great to give, these yearly updates on the work that we've been doing. Today, I actually, wanted to talk a little bit about the stabilization method that we've developed. So far, it was a lab, batch system that we operated in the lab, and we're sort of starting to think of how does this get scaled up and get used in source separating systems. So before I do that, though, I do want to, acknowledge that, much of the work that I'm going to present about today was initiated by Phil Arvey, who was a PhD student, graduated last year, and then currently Ehsan Bute, who is a PhD student who's also working in the lab. The other big acknowledgement is to Prithvi Simha and Dylan Randall. I think it was through a presentation I did at the summit maybe 4 years back that I got introduced to their work, but then Prithvi reached out, and actually we ended up collaborating. This last summer, I ended up spending some time at SLU in Sweden as part of the August T. Larson Guest Researcher Program, which developed a lot of the ideas that I'm going to talk about. Initially, actually, the funding for what I'm going to talk about was supposed to come from the EPA. This was basically a design project that I had pitched to the P3 program. Unfortunately, the program, I think, is now canceled. And so we lost the funding, but we still continued to do the work. And so, much of what my lab does is try to look at how electrochemistry can be applied within the context of wastewater treatment. About 7 or 8 years back, when I first got introduced to the field of source-separated urine and nutrient recovery, I kind of figured, let's try and find some problems where maybe some electrochemical approaches might be of use. And it was stabilization, where we thought that there is potential use because we thought we could in situ produce chemicals that are needed for stabilization rather than relying on buying those chemicals, storing large quantities, at a restroom scale or a building scale. And so the idea was, can we come up with a method where we produce a chemical of interest for stabilization in situ as needed at the concentrations that are needed. The great advantage with electrochemistry is that you also don't need to usually apply a lot of power to produce low concentrations of things. And so our idea was, well, hydrogen peroxide. Or how about try hydrogen peroxide as the chemical that would stabilize urine? So it would inactivate the urease enzyme, stop any hydrolysis of urea, but potentially also, residual concentrations could ensure there's no microbial growth over a long time. So the idea is source-separated urine goes into the cathode of an electrochemical cell. And so we are doing cathodic hydrogen peroxide production. Turns out you can also anodically produce hydrogen peroxide. But what we are doing is we are reducing oxygen to hydrogen peroxide. When you combine with anodic water oxidation, there is an input voltage theoretically necessary, but it's small. And again, if you're not needing to operate the system at high current densities, you would end up not needing a lot of power input in such systems. And so, this is a schematic that shows the concept, but essentially, what we are imagining is that you would have electrochemical cells that look something like this. So this is sort of like a 3D rendering of a cell, something that's made with pretty cheap materials. The electrode materials actually don't need to be really expensive either. So hydrogen peroxide production is catalyzed on carbon electrodes. Because you're low current, you don't need any fancy, precious metal catalysts on the anode either. And so you'd end up with some sort of a cell, which would, be these, modular cells that you could put in, at the back end of a restroom or a source separating toilet. We did, I've presented about some proof of concept results before. We actually have published results on how this works as well. But I actually wanted to show an unpublished result from a batch experiment. This was done a few years back, but we were trying to publish our first work on this. And one of the comments we got back from reviewers was, well, you should really show this with real urine. I think we were able to, sort of sneak the paper in still without doing the experiment. But then we did eventually do the experiment, which was we would take these cells and feed real urine into these cells. These are still batch experiments. And the idea is that you would over, fixed duration pass electrical current, produce hydrogen peroxide inside the urine, and then track over time what happens to urea or ammonia. This experiment, we actually did one up. We just didn't do ammonia measurement. We developed a method for urea analysis and tracked urea. And so the three data sets that you see here are untreated, which is we add urease to real urine. You obviously see a decrease in urea pretty fast. We also did an experiment where just hydrogen peroxide at the kind of concentrations that we can produce in these electrochemical cells. We added that, very little urea hydrolysis, and then we did pass real urine through the electrochemical cell. Again, batch experiment. So this urine stays in the cell, then we take it out, and then we track over time what happens to urea, and very similar to just chemically produced or chemically added hydrogen peroxide would be. And so the advantage here is that we don't need to store any chemicals on site. The proof of concept, the electrochemistry works, and that you are able to stabilize urea for a long duration. The next step after this was, yes, okay, how do we then incorporate this into a source separating system, right? So the idea is, yeah, we can show it in the lab. We can add, some volume of urine in an electrochemical cell and show that it works. We started brainstorming some ideas on how this would work. And so the concept that we came up with was that we would have source separated urine go into a tank, and the contents of the tank are actually what are recirculated with an electrochemical cell. The reason why we figured this was going to be the best option, there's obviously an option where all of the urine as it comes in passes through the cells and goes to a storage tank. But we were worried a little bit about precipitation potentially on the surface of the electrodes, because you are slightly raising the pH when you do the production of hydrogen peroxide. So you kind of figured a recirculation tank would also serve as sort of like a seed precipitation tank. So any precipitation would actually be removed in the recirculation tank itself. And so the idea would be source separated urine comes into this tank, we have a recirculation with the cell, and then we can get stabilized urine out. And we can maintain, on the basis of how much new volumes coming in, if we maintain a certain concentration of peroxide through on and off recirculation, we would be able to, have stabilized urine at a residual peroxide concentration go out. So we wanted to perform an experiment to do this. And so the experiment that we did was we did this twice, and I'll show you both of the results with synthetic urine and real urine. But you're sort of replicating like a institutional building with a source separated system where 8-hour workday. We had one experiment where we added synthetic urine every 45 minutes, so simulating a urination event for an 8-hour workday. So 45 minutes, every 45 minutes you add new volume for 8 hours. It turned out over a period of the 8-hour day, we would then end up adding 3 liters of new urine into this recirculation tank. During those 8 hours, the recirculation continues. In this particular experiment, we had 2 electrochemical cells, 15 centimeters by 15 centimeters. So that's 225 centimeters squared. And the idea was we would then, at the end of each day, collect samples from the recirculation tank, and we would then add urease just to see, what sort of stabilization we would get from the residual peroxide in there, and we would track hydrolysis in storage for over 2 weeks. So in the next couple of slides, I'm going to show you the 2 experiments, one with synthetic urine and one with real urine. But this is kind of, the photograph here kind of also shows the circulation tank along with the 2 electrochemical cells that we had. So this is the first experiment. I mean, we did a bunch of different experiments to lead up to this experiment, but this experiment is where we had synthetic urine every day. We are adding 3 liters periodically while the urine is circulating with the cell. At the end of the 8 hours, the circulation stops. Then when the recirculation stops, we collect a sample, add urease in it, and then track over time. Now, the first graph on the left shows hydrogen peroxide, which is great here to show that once we produce a certain hydrogen peroxide, it actually stays there for a long time. Now, this is synthetic urine. You'll see in a second in what happens with real urine. But what you also see is that each different color here represents the next day after. So at the end of day 1, we had about 200 to 300 milligrams per liter. At the end of day 2, we raised it to 300 plus, to 400 plus, to 500 plus, and so on. We actually, what I'm not showing on the graph here is that we actually can push the concentrations to as high as about 2,500 milligrams of peroxide per liter. So a steady state operation could actually even happen as high as 2,500 milligrams per liter. The more important graph is the one on the right, which is we are tracking ammonia concentration every day as well in the samples collected from each day. And you can clearly see that the control, we see a sharp increase in ammonia reflecting the hydrolysis. But samples from day one, when the peroxide concentration is about 200, there's some hydrolysis, but not too much. I mean, you go into a few hundreds of milligrams of nitrogen per liter, but not higher than that. But apparently, it seems as if 500 may be the magic number. So above 500, you see very little hydrolysis, at least in synthetic urine. Now, this is synthetic urine, so we obviously wanted to redo this with real urine, and so we did do it. Where the key difference here is that on the peroxide side, you do see some drop in peroxide in storage. So this is maybe the organics in urine reacting with the hydrogen peroxide. You see slightly higher ammonia concentrations to begin with, but then you also see the same kind of result where if you had a residual concentration of about 500 milligrams of peroxide per liter, you do end up with fairly stable urine in storage, again, for very long durations, right? So all of our sort of stored samples we were keeping for 2 weeks to make sure that we were capturing if there's any hydrolysis that occurs later durations. No microbial growth that we can observe in any of these samples as well. And so I kind of wanted to leave with a few take-home messages from our work. We have shown multiple times that electrosynthesized hydrogen peroxide can prevent in situ urea hydrolysis. This is now done with a system that is configured at least in a way where we would envision how this would work in a real system. With both synthetic and real urine, I think the second more important, with real urine also, we were able to show that hydrogen peroxide concentrations, as long as we get to about 500 milligrams or so per liter, they are enough to prevent significant urea hydrolysis. I stayed here continuous. I was always thinking of whether I should use the continuous term because it's not as if the urine's coming in continuously. It's a system that's being operated continuously. With real urine, we do see hydrogen peroxide residual concentrations decrease with time. So there is potentially further work that we have to do with variations, more longer time operation to see if variations can still allow us to maintain those kind of residual concentrations. And finally, we can actually produce all the way up to 2,500 milligrams per liter. So system, what I was showing in the earlier two graphs is basically system startup. But once you are operating it continuously for a long time, you may be able to maintain, 2,500 or more milligrams per liter of peroxide, which would ensure that you have, stabilization, and which would ensure, even with variation in the composition of the urine coming in, stabilization, because you wouldn't use up all of that hydrogen peroxide for other oxidation reactions. So with that, I'll stop, and I'm happy to take any questions that you might have. Thank you so much.
Thank you, Dr. Popat. That was great. We have one question so far in the chat. It says, could you please share how the pH variations differ between these two cases, with and without peroxide?
So great question. So when we were running our system in a batch type setup, we would always see, because peroxide production consumes protons, the pH go up. And that would lead to all sorts of precipitations. We saw a whole bunch of calcium phosphates that would precipitate. In our current system that I showed, we actually figured out a way that if we kept the anode pH very low, we actually have a lot of protons moving from the anode to the cathode through the CEM. So you don't actually see a whole lot of pH increase. Our synthetic and real urine were both similar to around 6 or so. And by the end of the recirculation, at the end of the day, we were measuring maybe 6.5, 7 or so in the pH. We did still see some precipitation in the tank, no scaling on the electrodes, which was great, because in our earlier studies, when we were doing batch studies, there was always scaling on the electrodes. But yeah, the pH goes up, but not by a lot. I mean, 7 was the max that we noticed. Went up a little more in the real versus in the synthetic, and I think that's the buffering effect of the synthetic, whatever buffer we were adding.
Oh, that makes sense. That's very neat. We had one more question. We have time for a couple more questions because I think we're only going to have one more presentation in this section. So we have that. The good news is we have time for some more questions. Another one here is, can you comment on the energy required to produce a given mass of peroxide?
Yeah, so great question. I didn't include a graph on that, but we can actually compute the kilowatt hours that are needed per meter cube. What I think I had on one slide, and I should have pointed it out, is that, so theoretically 0.69 volts in practice, you have inefficiencies in the system. We end up typically applying 2 to 4 volts, but our currents are in the few milliamps, right? So this system that I was trying to, we were trying to replicate as one event every 45 minutes for an 8-hour day, we were passing electrical current at a few tens of milliamps, right? So you multiply that with, the voltage, it's really small milliwatts that was passed over this system. So we did try to normalize to meter cube, and again, it was really small to even be of significant. I actually had a capstone design team that I was advising as part of a capstone design class that I was teaching, where they came out and said the electricity cost for a single restroom would be about $12 a year.
That's a nice that's a nice way to communicate that. Yeah, for sure. That's great. And then we have one more question from Rebecca Nelson. Can you comment on the idealized cost? You kind of talked about this just a little bit right now, but of peroxide versus acid or base for urea stabilization.
So that's a great question, because what I did mention was just the operational cost, right? So
Right.
buying peroxide in a highly concentrated form and storing it versus producing it in this particular way, it turned out the operational cost was going to be way cheaper with the electrochemistry. There is a capital cost. I think we found, again, with that capstone design project, we found that it was about 6 years or so is when it's the payoff period for the electrochemical cell, where
Right.
so about 6 to 7 years is where it was ending up as. And now we can try and make the cells cheaper. It is the membrane, which is the most expensive part of the electrochemical cell. So we can find cheaper membranes. The thing is, we were using Nafion in all of our studies, which is a more expensive cation exchange membrane. So maybe there's a way, we haven't yet tested there are cheaper versions of cation exchange membranes available. We haven't tested those yet, but my guess is they would work just as well.
Awesome. Well, thank you for the presentation and for the thoughtful answers to all those questions.
Thank you so much.
Great to have you. Also, I should just introduce myself real quick, because I kind of jumped in here without an introduction. My name is Arthur Davis. I'm the operations director here at Rich Earth, and I'm going to be moderating this next little section of the summit. So, our next presenter is Chibambila Simbeye, who is a PhD candidate in environmental engineering at the Swedish University of Agricultural Sciences, SLU, specializing in sustainable sanitation and nutrient recovery. His research focuses on developing scalable, decentralized urine dehydration technologies to reduce environmental pollution and support circular systems. Chibambila believes that sanitation is a fundamental human right, and he is committed to advancing inclusive, source-separated sanitation solutions worldwide. And there you are. And if you want to share your screen and take us away, that'd be great.
Okay, great. Let me see. Can you see my screen?
Yes, although
Is it the
yes, there it is.
presentation?
Yes, we
Okay,
see the
perfect.
presentation. Thank you.
All right, thank you. Hi everyone, and thank you for the nice introduction. So I think my presentation kind of connects and follows up on the conversation started by Rebecca and Sudeep before me. So I'll be talking about urine stabilization, specifically with calcium peroxide. So please ask questions, and I hope you find it interesting. Yeah. Okay. So yeah, so just some background. Usually when you stabilize urine with any alkali, most commonly calcium hydroxide, the goal is to get the pH above 10. But as discussed earlier, when it's exposed to air, you have absorption of CO2, and that leads to formation of carbonates, which makes the urine system acidic, and the pH drops below 10. And then when the bacteria produce more urea, you have hydrolysis, which will result into the production of ammonia. And because of the high pH, you lose the ammonia as a gas. So this is something that's a problem, and we try to avoid it. So one of the proposed ideas was, what if we kind of used alternative stabilization? So like Sudeep proposed with peroxide that uses advanced oxidation to denature the enzymes. And we kind of got the idea from the modified Fenton reactions in wastewater treatment plants, where they dissolve a metal peroxide to produce hydrogen peroxide. So our hypothesis was that by using calcium peroxide instead of calcium hydroxide, you could have some form of dual stabilization mechanism where the pH is raised above 10, and then you also have the oxidative inactivation of urease, such that if you have residual peroxide in the urine, as Sudeep previously talked about, it inactivating or preventing hydrolysis, then even if you have this absorption of CO2, you could still preserve the nitrogen. So that's what we hypothesized. And then, so the objective first was to assess the dissolution kinetics, whether we could actually dissolve this calcium peroxide in urine. So we chose for, after a couple of pre-trials, of course, we decided to go with 4 major concentrations, which was 5, 10, and 20 grams per liter. And we also measured the pH, the electroconductivity, the dissolved oxygen, and of course residual peroxide, then also ionic species that could be prone to precipitation and could indicate something happening in the urine. So the same graphic again, when you dissolve calcium peroxide, you can get calcium hydroxide and hydrogen peroxide. But then, yeah, I'll come back to that later. So we collected about 10 liters of urine, and then we split it up into these container reactors where we performed the experiments in triplicate, and then we made the control, and which was basically calcium hydroxide. And the reason for this is when they produce calcium peroxide, it's produced as a mixture of a hydroxide and the peroxide in varying percentages. So most common is 50% hydroxide and 50% peroxide. In our case, it was 75% peroxide and 25% hydroxide. So we had to add a control of just calcium hydroxide that represents the hydroxide we're adding when we dose with the chemical peroxide that does have some calcium hydroxide. So then we sampled doubling the sampling for every minute interval. So we went 2, 4, 8, 16, 32, 64 minutes until we had like a full 24 hours of sampling. And of course, like I said, we measured these specific parameters of pH, EC, DO, and residual peroxide. We used titanium oxysulfate and calorimetry at 405 nanometers to see the absorbance, and then work that backwards to see how much residual peroxide we have in the urine. Then, of course, we then stored the urine, and the rest was used for the component analysis. That's with the ionic species like the calcium, magnesium, phosphate, and the ammonia. So to the results, the first is the pH of the urine. So for the different doses, you can see how the pH changed over 1,044 minutes, which is basically 24 hours. And as you can see from the graph, that the higher dosages had like an immediate spike in pH. And that's mainly because the more you dose at the beginning, it also means the more calcium hydroxide you add. And because of that high pH, you then also affect the solubility of the calcium peroxide. So consequently, for these 3 high dosages, we could not detect any residual peroxide. But interestingly, for the lower dosed urine with the 5 grams per liter, we did detect some hydrogen peroxide produced at the beginning. And then if we look at that graph in terms of detection, what it looked like, we'll see that the residual peroxide was only present in the first, I would say, first 10 minutes, and throughout the rest of the reaction, we could only detect changes in dissolved oxygen and also an increase in pH. So the increase in pH tells us that the calcium peroxide is dissolving, and also the increase in dissolved oxygen tells us that it tells us potentially two things. One could be that the peroxide produced is being degraded, and the other could be that the alternative mechanism, which is instead of production of peroxide, rather it produces oxygen, is happening. So in this particular beginning, we can see that what we have is the first alternative, which is the calcium peroxide breaks down into peroxide and calcium hydroxide. And then over here, we can see that the reaction that's possibly happening is that we are producing oxygen instead of peroxide. So it shows that the dynamics by which this dissolution happens are kind of complicated because you have like firstly two competing mechanisms, and then the stabilization you desire via pH control also affects the solubility of the calcium peroxide, which makes it dissolve much slowly. And then at the same time, the higher you raise the pH, the more unstable whatever peroxide you produce, if you produce any, gets. So then into some other results in terms of the component analysis. If we look at the calcium, we can see, of course, that in the control, which is basically just to repeat calcium hydroxide, the dissolution is kind of immediate. So you have this sharp spike in calcium concentration that just fluctuated slightly and remained stable. And then for the peroxide, because it kind of dissolves slowly, you have this gradual increase in the calcium concentration. And overall, we found that at best, you could only dissolve 17% of the 5 grams per liter. Then we can also observe that from the pH, which corresponds with the dissolution. So as it slowly dissolves, then the pH starts to increase and goes above 10. Whereas for the control, which is just calcium hydroxide, the stabilization is almost instant because it's much more soluble in that sense. Electrical conductivity was fluctuating but mainly steady. And what's also interesting is the phosphate, because we're adding so many cations. The phosphorus will obviously be precipitated when you have this saturation of calcium within the solution or within the urine. So you can later capture that as a solid fertilizer in however you design your process. Also interesting was the magnesium, and the reason in here is that in the control, the magnesium remains steady. It drops a bit, but remains steady because there's this immediate saturation of calcium. So the preferential precipitation is that of calcium phosphates because they're in higher the calcium is in higher concentration than the magnesium, where because the calcium peroxide mixture dissolves slowly, it allows for the magnesium to take advantage of that and then precipitate as some form of magnesium phosphate, most likely struvite, because when we look at the ammonia, we can also see that in the control it was steady, whereas in the calcium peroxide stabilized urine, the ammonia goes down, the magnesium goes down, and that could be because you also precipitate in struvite. So your mixture of solids at the bottom could contain struvite. All right, then going into the next point, some conclusions and implications of the study. I think the first thing is there is potential to have the dual stabilization of urine by calcium peroxide. However, it's within a limited window, and you have to really assess whether the benefits of the process kind of benefit what you're trying to achieve. So preliminary, we've just done like a dissolution study to see how well it dissolves and whether there's peroxide produced, which shows that yes, there is some, but albeit in small quantities. So there is potential, but limited with more optimization needed. Then, like I said, it's because there are multiple systems competing. You want to achieve a high pH for stabilization, but this will limit how much calcium peroxide you dissolve. And then at the same time, this reduces your hydrogen peroxide release and probably stability. and it could also accelerate its breakdown. So the most likely conclusion from this is you have to do some form of dosage optimization, but the thing is that it could be a very narrow window, or the other possibility is that you might be want to do with very low dosages, and what that effect has on like passive stabilization and amount of peroxide produced has to be investigated. And yeah, like I said, it could be an alternative for stabilization of urine. So it's nice to have multiple pathways you can take because technologies differ depending on context. Like Rebecca said in her presentation, they couldn't go with acid because, of course, the soils have like lime. So maybe there is a context that the soils are acidic, sorry. So maybe there is a context that suits this particular type of technology, but of course more work has to be done with this. And in that regard, I think the next steps would be to assess how the activity of urease in urine is with the calcium peroxide stabilized urine. So the thing about peroxide is you can't really measure what's been produced. You can only measure what's left. So if you produce excess, then you definitely see that there's some left. So what we don't know is if urine sits for a long time with calcium hydroxide versus calcium peroxide, how does that compare in terms of the activity of the urease? So I think that's an investigation that would be a good follow-up for this study. And thank you for your time. That was the presentation from me.
Thank you so much for the presentation. And we'll wait a moment for questions. One question I had was, did you
Mm-hmm.
try this in kind of have you had any success using it as part of any other processes for dealing with urine at SLU, or is it mostly was a lab study?
I think it was a study of interest that followed up from the previous studies done on the alkali stabilization. So it's mainly
Right.
at lab stage. Like Jenna wrote in the conversation, we mainly lean towards acid dehydration of urine. But if it yields highly promising results, which so far it's a bit blurry, there is potential, but not for what we thought, because the initial idea was also like for if you have excess concentrations, then you could also use it for like active oxidation of organics. But the concentrations we got were not so high, so then the motivation to put it into a larger product is not that much high. So
Right.
yeah, yeah.
No, that makes sense. Well, thank you for the great explanations and thank you for the work. That's great.